Oke, baik Bu. Siap. Oke, baik Bu. Siap.
Ladies and gentlemen, our webinar is about to begin. Before we start, we would like to ask your cooperation to turn off your audio. Thank you. Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon for all of us. First, I'd like to introduce myself. My name is Medu Tami Putri, an academic staff from Faculty of Pharmacy, Universitas Indonesia, and I'll be your moderator for this webinar. Ladies and gentlemen, thank you for joining us today for today's webinar entitled Application of Metabolomics in Preclinical and Clinical Studies, Our Experiences. I'd like to greet our honorable guest, the Dean of Faculty of Pharmacy, Universitas Indonesia, Professor Dr. Apotekar Arya Nuar Magister Science, Vice Dean for Academic, Research, and Student Affairs, Faculty of Pharmacy, Universitas Indonesia, Dr. Apotekar Fadlina Kanisa Putri Magister Science, Vice Dean for Resources, Venture, and General Administration, Faculty of Pharmacy, Universitas Indonesia, Dr. Apotekar Sutrio Magister Science, Chairman of Board of Professors, Faculty of Pharmacy, Universitas Indonesia, Profesor Dr. Apotekar Hayun Magister Science, Chairman of Academic Senate Faculty of Pharmacy, Universitas Indonesia, Dr. Apotekar Iskandar Shah Magister Science, all managers, head of program studies, and coordinators, Faculty of Pharmacy, Universitas Indonesia. The honorable speaker for today's webinar, Professor Dr. Teh Laike from Faculty of Pharmacy, University Teknologi Mara, UITM, Malaysia. The honorable professors, lecturers, and all faculty members who attend today's webinar. And last but not least, I would like to also greet all audience and webinar participants. Welcome to the webinar series organized by the Faculty of Pharmacy, Universitas Indonesia. For your information, this webinar series is intended to broaden our knowledge of recent research developments and strengthen international collaboration between universities. Today's topic is about the applications of metabolomics in preclinical and clinical studies, our experiences, which will be presented by our honorable speaker, Professor Dr. Teh Laike. Before we start, let me explain the agenda schedule for today's webinar. First is the opening. Second is the speech from the Dean of Faculty of Pharmacy, Universitas Indonesia. And the third, we will have the group photo session. And the fourth, we'll go to our main agenda, which is the webinar session, followed by the discussion time. And last one, we will have the closing. So let's move on to the next agenda, which is the speech from the Dean of Faculty of Pharmacy, Universitas Indonesia. Professor Dr. Apotekar Arya Nuar Magister Science, the time is yours.
Okay. Assalamualaikum warahmatullahi wabarakatuh. Very good afternoon, Professor Dr. Teh Laike, our college student, and all webinar participants. It is a very good opportunity. At noon today, we discussed the interesting topic of metabolomics. Today, we have a guest lecture from UITM, Professor Dr. Teh Laike, Professor and Deputy Director of Integrative Pharmacogenomic Institute from UITM, Malaysia, who has expertise and experience in the field of metabolomics. Thank you for accepting our invitation to give this guest lecture in this webinar. Metabolomics is a field that is growing very fast in various applications. Metabolomics, defined as the comprehensive analysis of metabolites in biological specimens, is a promising new technology to inform precise medicine practice. Historically, small amounts of metabolic have been used to diagnose complex metabolic disease as well as monogenic disorder, such as inborn error of metabolisms. Current metabolomics technology extend beyond the scope of standard clinical chemistry technique and is capable of precisely analyzing hundreds to thousands of metabolites. As a result, metabolomics provides a detailed characterization of metabolic phenotypes and can enable precision treatment at a number of levels, including the characterization of metabolic disorders, underlying disease, discovery of new therapeutic targets, and discovery of biomarkers that can be used to diagnose disease or monitor therapeutic activity. With this webinar, I hope that we can gain experience from research conducted by Professor Telai Cake, so that it will be useful in research and clinical pharmacy services at University of Indonesia. Thank you. Assalamualaikum warahmatullahi wabarakatuh.
Assalamualaikum warahmatullahi wabarakatuh. Thank you for the opening remarks, Prof. Ari. Okay, ladies and gentlemen, now we are going to take a picture. Would you please to turn on the video and please one of the committee to guide this photo session.
Okay, all participants, please turn on your camera. We will take a picture. On count 3. We still wait some of participant here. Silakan dibuka kameranya, Bapak Ibu peserta webinar. Kita akan mengambil gambar bersama narasumber dan juga Dekan Fakultas Farmasi Universitas Indonesia. Baik. Baik, karena ada beberapa slide, maka pertahankan posisi Bapak Ibu. Kita akan mengambil gambarnya. Untuk slide pertama, slide kedua, slide ketiga, dan slide keempat. Terima kasih kepada Bapak Ibu yang sudah menyalakan kamera. Saya serahkan kembali kepada moderator.
Okay, thank you very much. And for information to all participants and audience, the certificate will be given to participants that 100% attending this webinar by filling the field attendance form and fill the satisfactory survey and also pass the post test. Okay, let's move on to our main agenda. But before we start, let me please allow me to read the Professor Teh CV. Professor Dr. Telai Cake obtained her B.Pharm with honors from the University of Nottingham, United Kingdom in 1995. She then completed her Master of Clinical Pharmacy and Doctor of Philosophy from University Science Malaysia in the year 1997 and 2002 respectively. Her research areas are experimental and clinical pharmacogenomics and clinical and experimental metabolomics. Currently, she is working as a professor at the Faculty of Pharmacy, University Teknologi Mara, UITM Malaysia, and as the Deputy Director of Integrative Pharmacogenomics Institute, or I promise, one of the Center of Excellence in UITM. She is the founding member and the president of the Malaysian Metabolomics Society. She is also involved in the Pharmacogenomics Global Research Network as the regional co-chair of the developing countries. All right, without further ado, please welcome our honorable speaker, Professor Dr. Telai Cake. Good afternoon, Professor.
Good afternoon, Dr. Putri, Dr. May.
Yeah. Okay, so now the time is yours. Please, Prof. Okay.
Terima kasih kepada organizer, terima kasih kepada Prof. Ari, and also Dr. Rani for actually inviting me for this particular seminar. Yeah. So I think without further ado, I just start with my slide presentation, and then after that, probably we can do some discussion on our further collaboration. Yeah. I would like to actually for the host to allow me to actually do screen sharing, please? All right. Can I have permission to actually share the screen so that I can actually project my slide?
Yes, you already have. You already can share screen, Prof. Teh.
Oh, all right. Now can. Yeah. Thank you so much. All right. Selamat tengah hari semua, hadirin semua, pak-pak, ibu-ibu. Okay, izinkan saya untuk present my slide actually or sharing session in English, yeah. All right. So yeah, actually metabolomics too is nothing new actually. Yeah, from this slide as what we can see actually, it actually have started in 15,000. 15,000 that time people already actually use the concept of metabolites actually to help to diagnose diseases actually. For example, in 1506, yeah, Ulrich actually has used different colors, smells, or tests of the urine, or even body odor actually to actually help him to actually diagnose diseases. Yeah, but of course, with the advancement of technology, then they come a very good separation, chromatographic separation techniques that was developed in 1960. And from there, actually, people start to advance further to use more quantitative approach actually to analyze the metabolites that give the color or give the odor, the bau to the urine or whatever that actually denote whether or not the person actually are having problems or having disease or not. So 1978, basically, the papers on quantitative metabolite profiling, based on gas chromatography has been published. So that is probably the first official paper that was being published. That was in 1970s. Yeah. All right. So basically, that is about history of metabolomics. So today, actually, metabolomics has been used in medical researches, plants and food kind of research, as well as production and also biotechnologies. So in terms of medical research, a lot of people like to use metabolomics because it is easy, an easy tool to use for screening of diseases. It can also be used to actually understand gene function as well as to identify therapeutic targets like what Prof. Ariel actually have said just now. So elucidation of the functional aspect, the pathology, disease marker discovery, and then toxicity all under medical research has been actually achieved quite a prominent kind of achievement under metabolomics. So for plant and food, basically people are trying to find out what metabolism pathway of the plant can be triggered to produce certain metabolites that could be beneficial as a drug or as an active ingredient that can be used for therapeutic purposes. Yeah. So for biotechnology, actually, certain metabolic pathway has been manipulated, to actually produce or increase the fuel production. Yeah. For example, from algae, people understood that it was due to carbon kind of metabolism pathway that we can manipulate in order to produce more fuel from algae, for example. So when we compare, this one is just to give some encouragement to us in Asia doing metabolomics. So globally, this is basically like, for example, the axis here is for global. So people have published up to like 12,000 kind of papers globally. But in Asia, the one in red, we only achieve, maximum of about 200 something in 2021. In 2022, we are the papers that published from Asian countries are only about 50 plus. So as a rough kind of a guide, we can say people publish 4, we only publish 40, we only publish 1 paper. So there is so much room for us to improve, to actually see how we can actually meet at least setanding lah dengan people from the other side of the country. So metabolomics basically, is a high throughput kind of analysis of metabolites. It actually simultaneously, multi-parallel, it measure levels of different cellular metabolites. It can be a very large number, like what Prof. Ariel has mentioned. It could be hundreds, it could be thousands, right? But if you're using LCMS, a lot of time, the metabolites, you probably don't know them yet. They are sort of new, but you can see them as peaks or profiles in your LCMS chromatography. Yeah. And generally, basically, we have two types of metabolomics that we work with. One is a non-targeted, we call it a global metabolomics. In this particular experiment, basically, you have sort of a model. You basically will just extract using a wide kind of spectrum approach to try to get as much of metabolite out from there. So it's global. And then you just detect using a machine, right? Then after that, only you make sense of what is different between the treatment or non-treatment approach. Yeah? Then you can, from there, you will do deduction or try to find pathway through the interpretation, yeah, when you do pathway analysis. But there is also another metabolomic approach that is called targeted metabolomics, yeah? Where you already know what metabolites that you actually wanted to see, or you already know what kind of metabolites that will differentiate the clinical condition that you want to study. So you will try to actually extract those metabolites out. Yeah. Then from there, actually you detect it, you calibrate your metabolite sort of MS machine to make sure you actually detect those metabolites. Then after that, you do interpretation. So a lot of time, these targeted metabolomics, you may actually use LCM as QQQ as well to actually detect and quantitate. Yeah. So this is basically the comparison between untargeted method as well as targeted method. For example, for the untargeted method, you can actually, it's unbiased. Basically, you do not know what you wanted to look at specifically because you are still trying to find. So you don't really have a hypothesis yet. Right? So for this particular method, basically, you will have a lot of complex data, and also you may not have repeatability because, even though we have been doing a lot of this untargeted metabolomics, we find that even from the same sample we inject 4 times, there are still variation of sometimes we detect certain metabolites, whereas another injection, we don't actually detect the metabolites. So our job here become complex in the sense we need to look into what is the repeatable kind of metabolites that is consistently present in our system. So we have to use a very good tool for that. Yeah. And basically, for this untargeted metabolite, you need to use a machine that allow us to have exact mass kind of identification. That's why we actually, for untargeted metabolomics, we have to use LCMS QTOF, because the QTOF, the part of the machine, allow us to identify precise, accurate mass so that we actually can annotate to match the metabolite to database. Yeah. If you just use TOF only, you probably may not get an exact mass kind of metabolite. You can get some masses, but the masses are not good enough. The masses are not accurate. So you actually have to use QTOF. Yeah. So that is basically the differences. And for untargeted metabolomic, because you do not know what you're looking for, basically it's general, it's unbiased. So it's simple processing. The more simple you process the sample, the better it is, because if you actually use a complicated methods in order to process your sample, then you may actually lose some metabolites. So for example, in our site, we actually just use methanol or ACN just to precipitate the protein, and then whatever is left in the supernatant, we will just concentrate it and actually inject. We don't do many steps of processing. We try to do it as minimal as possible. But however, when you're doing targeted, that will be different, yeah, because you want to recover as much of that specific metabolite of interest, yeah. So you may have to use different kind of solvent extraction or things like that, yeah. So there is something here that that was found. I mean, I actually was reading, and then there is something that new coin, pseudo-targeted methods now. So because there are pro and cons, there are advantage and disadvantage for untargeted and targeted. So now people want to match these two. Yeah, they wanted to get the best of these two methods. They want to make sure they can actually quantitate properly the metabolite. So they come with the pseudo-targeted kind of method. But of course, this means that you need to use another equipment. So in this pseudo-targeted, basically you need to use LCMS QTOF as well as LCMS QQQ that allow you to target actually. Yeah. So metabolomics basically as overall is a holistic analytical approach to the metabolism, not guided by specific hypothesis, right? And it helps to determine how metabolites level responds to genetics or environmental changes. So basically, when you design your experiment, that is what you wanted to look at. You wanted to look in genetic variation that has caused the differences in metabolism. For example, the inborn error in the babies, right? That was because of certain genes malfunction. That's why they cannot metabolize certain sort of the metabolism when haywire, or then there are certain metabolites that was detected and not detected, that kind of thing. And environmental changes, yeah. So that is basically the beauty of metabolomics. It can actually catch the differences that is due to genetics as well as environment that cause the metabolism to be different. And again, this is general approach of how we take metabolomics from the beginning to the end. From biological samples, it can be human, it can be animal, and then for human and animal, you actually can take fluids or tissues. Yeah, these are usually the two things that we actually harvest from our experiments. And you can also study yeast, you can also study bacteria. Yeah, you can actually look into from the bacteria, you can actually take the culture cell. For example, you can actually harvest only the bacteria cells using filter, right? When you grow the bacteria until 10 power of 8, yeah, then actually you filter the cell and just capture the cells, and you can actually study, break the cells and study, so then you get intracellular kind of metabolism. You can also actually sample out the growth media of the culture cells to look into what are the metabolite differences that have already happened, which is being released from the bacteria due to your experimental approach. So there are many things that we actually can do. Yeah. And then with the analytical, a lot of time now we actually using LCMS, because LCMS has a better and broad spectrum compared to NMR. NMR is more specific and it has a narrower kind of metabolites that you actually can identify. Yeah. So for the analytical profiling, what we have to look into is we have to identify the key features. The key features that is being recorded from the analytical profiling approach include the metabolites, different metabolites, the signal that proportional to the levels or the amount of metabolite. But we have to make sure it is precise and robust. Precise means that you keep repeatedly identifying or detecting the same metabolites most of the time. Right? Robust means that actually in machine, if you change machine, you still detect it, or if you change the operator, you also still detect it. So that kind of robustness, which we actually have to also validate. So in our experiment, a lot of time we put in a QC. Yeah? We put in QC. That is very important for us to monitor the machines are behaving correctly, and the sample that we actually injected to the machine are also of the correct amount and also has been treated or processed correctly. Yeah. Then after that, you can use different software yeah, that allow you to do statistical analysis, yeah, to differentiate what are the metabolite, what is highly presented or low or less presented in the different samples that you actually detect. So that is basically a heat map kind of thing. Yeah. So you can use univariate or multivariate statistics and also do some correlations. Yeah. And again, after that, you can do cafe analysis. How you want to do this? Don't worry, actually, because there are already free software that is already made available for us, yeah, who wants to do metabolomics. Yeah? So from there, you actually can identify disease marker, signature of the disease. You can actually also look into the patterns of a metabolite phenotype. You also can actually look into drug activity and efficacy, and of course, discover new biological mediators, yeah, from this after you have done the statistical analysis. So that is basically a general thing. And then I promise, basically, we have been using metabolomics to do both preclinical and clinical kind of analysis. Yeah. So for preclinical, we have been using metabolomics actually to understand better what are the biological pathways, yeah, of new compounds or basically extract, yeah, crude extract that we have been investigating. For example, maybe Kachet Fatima, we want to see that what is actually the function of Kachet Fatima in treating people with menopause. I don't know, yeah, to make the person young. Right? So we can actually do in cells first, then after that we can actually collect wanita punya blood samples and look into whether the extract actually helps, how they help, yeah, how they change the metabolism of an old woman to become a young woman, you know, those things we can actually do. Yeah? And for clinical, we also has been actually using metabolomics to actually understand better pharmacogenomics. We know that pharmacogenomics basically is a study of looking into genetic variations, yeah, in changing, in causing adverse effect, in causing different kind of drug effect we see in our patients. So we want to see how these different genotypes actually put in their effect, how these different genotype actually determine the effect from different patients. So we can actually also, if we have a good pool of patients, we can actually genotype them. For example, genotype them with gene A, then we can divide them to gene A homozygous, gene A heterozygous, homozygous mutant. Then we actually profile, you know, after giving a drug, what happened in terms of the metabolism of this patient with different genotype groups. Also, if we can actually, I think that would be a very nice project to be done with Indonesia, because you have a big pool of subjects actually. And then we also look into biomarker for diseases. For example, you know, we have cardiomyopathy patients who have heart attack. Then we want to see what are the markers in these heart attack patients that we can actually differentiate it from healthy people. Probably we can also later use that as a biomarker to actually, you know, we take somebody's blood, you know, and check, okay, this person actually are on the way to become to have attacks. So then we can change the diet, we can change a lot of things based on the metabolism. That is sort of like not good anymore in the patients, because there are a problem in terms of the food that they take. Remember, metabolomics can also identify the environmental factors that is different in different people. So the food that you take, the exercise that you do may change the metabolism of the patient, and that can be reflected in the metabolite profiles that we actually see in different patients. And that profile can be used to help the doctors to actually say, okay, you know, whether you have exercised enough, you have been eating correctly or not. So those things actually, yeah. So why actually metabolomics has been used? Yeah? Because metabolomics or the metabolites actually are not like what do you call it, proteomic or transcriptomic, whereby, you know, they reach a plateau like this. A lot of time metabolomics are in a straight line relationship, yeah, in the sense that if the environmental change, then the metabolic profile will change. So that is why metabolomics has been a better approach in order to monitor to the extreme ends. It has a broad spectrum in actually analyzing or trying to correlate diseases as compared to transcriptomic or proteomics. Yeah? So because metabolomes are a step nearer to the actual function, yeah, as you said here, rather than proteomic or transcriptomic. Okay. All right. So in I Promise, that's what we do. All right. We collect from samples of subject, of plants, of cell culture, yeah, anything from this animal, the rats. Then actually a lot of time we collaborate with people. So if our collaborator are at a distant site, we will usually ask them, can you snap freeze your samples? Snap freeze so that we stop the chemical reaction that is happening in the tube, right? Then only when you send it to us in the lab, we will do processing. Yeah, the snap freezing it is important to make sure you freeze scan the reaction, no more reaction happening, because you can use what do you call it, liquid nitrogen to do that. Yeah. Then after that, you can actually process it. You either centrifuge it or you actually extract it. Yeah. So very simple. Here we actually do just spin down. We add some chemical solvent, then we spin down the proteins. With that, then we actually extract the supernatant. Then actually we would dry it and inject it into the machine that we have in our lab. Yeah. So this is basically Q-top. You see a chimney here. We actually using only HPLC that is linked with an MS Q-top MS, LCMS Q-top. That is what we call it. And for this LCMS Q-top or metabolomic radars, these are the metabolites. Usually we can quantitate, or we can actually relatively quantitate using LCMS Q-top. So these are the small molecules, peptides, oligonucleotides, amines, amino acids, lipids, alkaloids, but not DNA lah. Yeah, we don't actually quantitate DNA here because these are the small molecules. Then actually when we do this analysis, we have to have quality control. So we actually have reference mass. What it means is that at every 1 minute that the sample are being profiled, the machine will inject reference mass and they will do automatic correction so that the mass differences is reduced. Yeah? Then we also inject separately pool samples. Pool samples means here that, for example, in a study, you are actually doing 100 subjects. You will aliquot certain amount of their serum or their plasma or whatever, you aliquot out, yeah, so that you can actually pool the 100 sample into one tube. Then from there you aliquot out, you will extract that pool samples with each batch of the sample you analyze. Because after that you will have to do comparison as in whether or not the assay you do are actually being standardized. Yeah? And in iPromise, we also have this, we are lucky to have this package that is called MPP, Mass Profiler Professional. That is the basically a statistical package as well as analysis package by Agilent. Yeah? So after we do this sort of sample analysis, we actually can put into the software, yeah, the different treatment kind of samples. Then this is what they will give us. They give us heat map. They will compare the different metabolites, you know, and the treatment groups, and then they will tell us, for example, okay, these metabolites are high in this treatment group, whereas low in this group, and high again or orange color means medium in this group. Yeah. So you can look into in this 3 treatment group, for example, these 2 groups have high expression of the metabolite, whereas this group have low expression. So from there we can already see very clearly from the heat map. Yeah. Then after that you can do some more analysis using other pathway analysis. Yeah. And you also can actually do target metabolomics lah. For example, you have certain metabolites that you actually know the quantity, you can actually quantitate the levels, for example. Yeah. So beside, if you do not have the MPP, don't worry. Yeah. You can actually go into metabol analysis. Yeah? Metabol analysis have also free, basically it is a free software that allow you to actually do all this, just same as MPP actually. Yeah, for you to do all this peak detection, meaning you can actually detect the compounds. Yeah, you save your data in .d file, then actually you convert it to the format they want. Then you can actually do all this analysis. Yeah. You can do peak alignment. Then after that, they will actually filter out noises for you. They will do normalization for you. Then again, they do statistical analysis for you, univariates, feature selection, cluster analysis, classification. Then after that, they can allow you to go, to do pathway mapping. If you're connected with KEGG, they will tell you this metabolite you detect actually is located in which pathway. So that pathway are actually sort of not abnormal in your samples. Then actually they can give you a lot of things. Actually, you can actually go in there to have a look. Yeah, and they have tutorial for you, so don't worry actually. Yeah. Then I think in 2021, actually this metabolomic workbench have come up with a very sort of schematic kind of assay for analysis as well. You know, for example, they allow you to actually upload your data into their database and they will do everything for you. Okay, you just have to describe online what is your project, give the name to your project, and then you actually provide them your study design, your treatment and everything, and then you give them the raw data. They will do the analysis for you, and it can be NMR or it can be MS based, and then after that they will provide you results. And it's basically less than 15 days. You upload the data, they will provide you the analysis. Yeah, in less than 15 days, actually, they will give you the data. So don't worry, people has made things very easy for us. Yang important now is we need to have our study design. Yeah, our study design should be correct so that we actually analyze it. We have to collect the sample correctly. The sample storage. Yeah. You have to fish dry it properly, and those things actually, those steps will actually affect the result. As we always tell the students, rubbish in, rubbish out. Yeah? So if you do not do it correctly in the first step, the analysis that people do for you later will be rubbish. Yeah. So So this human metabolome database, they already keep improving. They keep improving the database. So inside the HMDB, they actually have now like 2,000 over drugs as well as 800 drug metabolites. Yeah, and then they also have other subs database, you know, like for toxin, environmental pollutants. So if you are an environmentalist where we want to look into pollution, you can also actually use metabolomics. Yeah? So there are also in the database 220,000 metabolites. Just imagine if you have samples that contain 220,000 metabolites. Yes, HMDB already have those metabolites being cataloged, so not to worry. So if you want to develop a database, what database you want to develop, please make sure it's different from these 220,000 metabolites. Yeah. So probably because our part of the country is different, we have plants, we have new metabolites that this HMDB do not have, then probably it's good to do it lah. But if you want to develop database, then we have to look into what people already have. Yeah? All right, and it's very nice under metabolomic workbench. Yeah, people are moving into. Sorry, I mean you can't see. It's actually omic analysis. Yeah, if I can actually remove this, how do I remove this? Oh, okay. So this actually is omic. They actually have another platform where you can actually analyze multi-omics. That means you don't actually submit only metabolomics data, but you also submit genomics data. You also submit transcriptomic data. You also submit proteomics data for the same experiment actually. Then they can actually do the analysis for you. Just imagine that is how complex this is. It's no longer 2D or 3D. It may actually 4D or 5D. Yeah. So that is how complex when you actually visualize the different features of proteins, RNA, DNA, whatever that is inside there together with metabolomics. So that is the way forward. Yeah. You know, there is a lot of network correlation already available. Yeah. So metabolomics is only one. It's not everything also today. So okay, that is basically what we have got. I mean, worldwide, globally, that is what people have done. But I promise, I would like to just share with you what we have done. Yeah. So we have in this particular sort of discussion or today's meeting, I would just like to show what we have done for what do you call it, Mutinja Karubura, how it is being used as a liver tonic, and then what kind of pathway it actually has been profiled to actually have that effect. Then I also actually will show you one of our study on moringa leaves, yeah, where how we use it as an anxiolytic agent. And then for the clinical aspect, I would actually share with you some study that we have done on the metabolic changes that is due to myocardial infarction and how we actually produce a prediction model. And also, we have done quite a bit of clinical study on cancers, yeah, because there's a lot of metabolism dysregulation that happen in cancer patients. So we actually do some of this. So I would like to share some of this. And these two studies, as I said, we actually look into the effect of Mutinja Karubura against liver toxicity that has been induced by this CCl4 tetrahydrocorine that actually cause liver toxicity. So we want to see if we use Mutinja Karubura, can it actually prevent liver toxicity, right? And what happened? Yeah? And another study I want to share is actually the use of zebrafish also. We actually, this one is easy lah. We just take blood from rats and then we do metabolomics. But for this one, we actually use the brain of the zebrafish, which is very small, to actually understand the metabolomics. Yeah. This zebrafish was exposed to a lot of stress, yeah, over 14 days, and then we want to see what happened to them. Oh, yeah, sorry. All right. So this is the infographic. Yeah. For what we did is we actually identify the plant Mutinja Karubura in our country. Then we actually extract it. Then after we extract, we actually do some phytochemical screening, and we found that this plant extract actually contain flavonoids, tannins, tryptophan, and steroids. So look at all this. You know, if what do they actually cause? I mean, what is the effect of this particular compounds? So what we did is this poor thing, we actually induce it. We inject CCL4 to these rats and then make it liver toxic, right? So at the same time, we have groups of rats. One group actually we don't actually give this extract to the rats. We just cause it the toxicity. Then another group that is nothing, yeah. So after that, we basically harvest the liver and we look into the liver effect. And of course, those with CCL4, you can see that there are inflammations. Poor guy, right? The liver is already affected. Then for this rats that is treated with this MCME, methanol, Mutinja carubura methanol extract, you see the liver is so nice. So from the histopathology, we can see that yes, Mutinja carubura actually prevent liver damage that is due to CCL4. But what happened in the metabolomics? So we actually do this metabolomic analysis using our CMS Q-tool. And from the PCA analysis, the statistical analysis, we can see that there are differentiation in terms of those rats with liver toxicity and then after treatment with Mutinja carubura. And then we identify actually some biomarkers that has been sort of expressed differently in these rats that is toxic, not treatment, and rats that is being treated even though they are also having liver injury. Yeah. So the major pathway that we found, it was sort of like affected during toxicity, as well as actually Mutinja carubura actually affected is actually those affecting the liver, liver function, as well as the what do you call that, inflammatory function. So in short, actually what we did is actually we do a pathway analysis. So this axis is basically more on whether or not actually it's significant or not, yeah. And this is basically the pathway impact. Pathway impact basically was a calculation of all the metabolites that is calculated together to see what is the impact of that metabolic pathway on that particular experiment. So different pathway, different experiment will have a different pathway impact. Yeah? It will not be the same. But what we want to see is usually this pathway, for example, it has quite a major impact in what we see as in the differences in the metabolites that we have identified. But you can say that it is not significant. There is an impact, but it's not a significant impact, right? And then this pathway basically, it is significantly different, but it has not enough impact. So what we can see from this is probably maybe the sample size is too small. We have identified something significantly different kind of pathway, but it's not impactful enough. So that is probably because well, maybe our number of animals was not sufficient. Yeah? But you can see that when we actually only use these 3 biomarkers or 4 biomarkers, 1, 2, 3, 4. Okay, the area under the curve, basically this one is done to look into how specific is this metabolites in differently identified in different groups. Okay, we can again look into this chart. For example, this one is false positive rate, right? This is true positive rate. So what we want for this, for example, this xanthocin, for example, A, is it when it has a what do you call that, true positive rate here, right? You should have a low false positive rate, right? So what I'm trying to say is this metabolite marker, it has an area under the curve of 0.98, yeah? And at this particular point, okay, we can actually achieve quite high, significantly true positive rate of nearly 1, yeah? With 0 false positive rate here. So what it means is a good marker lah. When it is true, you can't actually identify it to be poor. Same as for here, okay. This particular compound C, it has if it is only about 0.7, it has a true positive kind of rate when it has a 0 false positive rate. But if you want to go to a true positive rate of here, then this particular metabolite also have a very high positive rate. So what we can say here is that this metabolite only is a good marker at 0.7. That is when it actually has 0 false positive rate. Do you get me? So we want to actually, if what I'm trying to say is a marker is a good marker if it has a high true positive rate, but false positive rate is low. Yeah. So for example, this guy, he has a high true positive rate, and at this point also, it has only very low false positive rate. So it is a good one. Whereas for this guy, okay, if you want it to be true positive rate of here, at this point also you have a very high positive rate, which is nearly 1. So you cannot say that this marker is a very good marker because it is only having a true positivity of 0.7, whereby it is a false positive of 0. Yeah, so that is what we have to look at when we actually do our rock analysis for each of the metabolite, and you can actually do it using rocket analysis under metabolic analysis. Yeah, so from here, then we look into the changes of level between different groups. So for example, there are 3 groups here. You can see that when, so xanthocin looks like it's increased when it is being treated with CCl4, but in normal it is low, right? Yeah, and this treatment with the extract managed to actually change it back to normal. So what it means is xanthocin could be a good marker to say that yes, there is an injury in the liver, and that damage actually has been reverted back to normalized back by this MCME group. Right, so that is what it means for this marker A, xanthocin. Okay. So that is what we do for this particular study to actually identify which pathway sort of being affected with when these rats being injured, and as well as when you give MCME extract, what happens. So these are the pathway that has been sort of improved or changed, or we call it pathway involved. Yeah, include purine metabolism, bile acid basically is the one in the liver, right? Another one is bile acid biosynthesis. Both are actually liver, because the liver is affected. Of course, the first one that is affected is bile acid. Yeah. And then arachidonic acid metabolism, basically that is more on inflammation. Yeah. So these pathways are affected. So the next study on animal that we did that I want to share with you is on zebrafish. Again, we actually do extract it. We extract the leaves, yeah, and then with the alcohol extract of 3 water and 7 alcohol, right? And then we actually look into the zebrafish. What we did is that we have different groups of zebrafish, group 1, group 2, group 3, until group 6. Yeah. So what we did is that we actually exposed this fish to 15 minutes of stress. And of course, we treat it with different kind of things. The first group is normal. Second group, we treat it with the extract 500 milligram. Then the another group, which is with 2,000 milligram. The group 4 is actually 1,000 milligram, and group 2 is basically stress without treatment. Yeah, and then group 6 is the one that has been treated with fluoxetine, the drug that has been used to treat stress, right? So then we actually expose it to stress for 14 days. Then we look into the behavior test for another 15 minutes, and then this guy will be actually capturing it all the time. Yeah? And then this is basically the results in terms of different treatments groups, how much time they actually spend. And from our study, this is basically normal. And then this one is stress group without treatment. And then this one is basically a group treatment treated with the extract, but only 500 milligram. And then the second one is 1,000. And then the third one is 2,000. And then this one is treated with fluoxetine. So we can see that this extract actually at 1,000 milligram already have a very good effect to it, whereby because the zebrafish will spend more time in the light zone when they are not stressed, yeah, or they will spend more time in the top zone when they are not stressed. So it looks like this moringa do have an effect to actually reduce the anxiety of the fish, yeah, but it's again dose dependent. Giving too low you have an optimum dose of 1000, and if you actually give more, actually the effect is being reversed. And again, we do metabolomic analysis. So this is the PCA that we actually did. We want to see that what are the metabolites, whether or not there is a difference between treatment as versus control, treatment versus stress. So if there are actually metabolites differences that is being expressed differently in different treatment group, you can see things like this lah. So what we want is, as best, kalau boleh, we want to see very different groups like this. Yeah. But however, what we can see here is probably because the samples that we collect, we can't really actually differentiate, because it's the brain, kan? We just take and it's the whole brain that we actually.
Makanya aku pakai masukkan.
So this is basically the PCA. Again, we do pathway analysis, but this one you can see is a bit more complicated in the sense that, you know, the significant pathway is here, purine, and it has quite a good impact here also, yeah, purine metabolism again. And then the next one, you can see that this phenylalanine, tyrosine, it is very impactful. A lot of metabolites that is being captured by this study is all from this phenylalanine and tyrosine being actually involved in actually brain kind of activity. I mean, the study sort of tell us quite good, yeah. It reflects very well because a lot of time when you look into CNS function, these are the metabolites that actually will be affected. Yeah. So a lot of metabolites that was captured was actually of this particular group. Yeah. But the significance level was not as purine. Probably what it means is that you know, the effect of the extract is still not able to reach very significant differences. That's why they can the pathway impact is not very high in significance, but anyway is already more than 10 power of -2. Yeah, that is 0.05, more than 10, more than, I mean, the P, log P is already less than 0.05. Yeah, when you actually have a log -10 of 2. My cursor, so it's up to here is 2, right? So it means that it's already 0.05. So it's already impactful enough. It's already significant. Yeah. And a lot of the metabolites that you identify in your study actually are of this particular group. Yeah. So when you look into this AUROC analysis, you can see this AUROC is not that good as compared to xanthosine, right? Because you can see that actually the impact, I mean, not the impact, the significance here is about 0.8, right? 0.7 something, you know, and then because you can see the false negative rate is only 0.2. All right. So if you, there are a lot of gray area means there. Yeah? The biomarker is not 100%. I mean accurate lah. Yeah. So, but you can see from here is that, you know, when we actually plot the metabolite of interest, yeah, for example, for this particular metabolite, you can see, you know, one of the reasons is not a very good biomarker. You can see it's like widespread. The standard error is so huge. Even though you can see there is a difference between these two groups, but this group very so high. Same as for here, you see the standard error is so huge. Yeah, even though there is a difference. Same as here, even though there is a difference over the red line, but again, it's overlapped so much. That's why it's not a very good biomarker per se. You can't really differentiate treatment versus non-treatment. Yeah? But what I want to show you is that, yes, if you do properly, the metabolomic would give you an answer. But of course, how good is the answer? It depends on your study design. Yeah. Again, it depends on the nature of your study. If you are looking into brain of the zebrafish, probably this is the best that you can actually identify, because we managed to publish this study in Ethnopharmacology, which is a Q1 paper also. Yeah? Next, for clinical study, I would like to go through quickly. I think I don't have much time. I already spent a lot of time. So it's on breast cancer. Because the reason we did breast cancer is because actually, you know, it is one of the most common cancer among the women. So what we did here is, of course, we have to get ethics approval. Then after that, for this particular study, we actually collect plasma from the patients. We also collect tissue, tumor tissue and normal tissue from the patients. Yeah? So we for the plasma, of course, we have healthy control. But for the tumor tissue, basically the patients themselves are the control, meaning that the doctors actually sample from tumor tissue as well as the normal tissue from the same breast, actually. Then we actually do a sort of extraction of the metabolites from the tissues, as well as from the plasma. Then while waiting for injection or analysis, LCMS Q12, we actually store the sample at -80. Yeah. So, and then we run through the LCMS Q12. Of course, each sample will have to run 4 replicates, and we also run full sample of batch just to make sure there is no variation from batch to batch. So for the plasma, for patient versus healthy, we use positive mode, whereas for the tissue, we use positive and negative mode. And then for the tissue, we also actually have staging of the tissue, either it's a stage 1, stage 3, or stage 4 kind of cancer. Then we actually do processing, data processing using differential analysis, compound identification, filtering, or normalization. All right. Then for the profiling aspect, we actually do principal component analysis, statistical analysis, also MEPA analysis. And then for the biomarker, we actually do ROC as well as also look into the PLSDA again. So this is what we did. And before we wanted to see which solvent is best to actually extract out most of our metabolite, especially for the tissue, right? We actually work on several kind of solvent. We actually work on acetate, hexane, dichloromethane, methanol, and things like that. So, and from here we can see that this ACN actually still gives us the best sort of coverage in terms of the different kind of metabolites that was identified. Yeah? So usually when you want to optimize something, you need to look into the polarity. Your sample, is it you want to extract more of polar one or what? Yeah. So you can actually have this polarity chart with you also. Yeah. And a lot of time before we actually do the whole study, we want to do a pilot study first. We just want to see which one is better. And for this slide, we actually look into the plasma of the patients as well as healthy. So the blue dots are actually patients one, the red one are healthy patients. You can see when we run the plasma of patients as well as healthy, the separation is very good. The PLS, the PCA is very good. That means that these two groups actually have already different metabolism that is happening in the patient's body. And these are all the metabolites that we have identified to be different in terms of for these patients. So we classify it according to fatty acids or lipids or vitamins, nucleic acid. And we actually see whether in patients and as well as in healthy volunteers, whether it's high or low in these different groups. So you can see some of this metabolite was low in patients, whereas very high in this patients group. You know, there are some high, there are some low here. So different metabolites actually show differently. But I usually will get my students to study more. Why is this high here and low there? Yeah? So that is what we're going to be able to answer. Then based on this metabolite that we identified earlier, we're going to do oral analysis. So we want to see which metabolites are able to actually give a very good sensitivity and specificity metabolite. Usually we say a cutoff point of 0.7 is already good enough, which we can actually include in our model. Yeah? Then after that, we will do a model. For example, in this case, we actually have patients. We divide the patients to two groups. This is what we did when we want to come up with a prediction model. So from this metabolite that has been identified to have good sensitivity and specificity, we actually put into this model, and you can see that healthy is is not a full 0. Patient is a full 0. You can see that they are again very good differentiated between patients and healthy. This one. So what we did is that after that, we actually did this as a model. This is already a predicted kind of probability. Then we collect another set of patient sample, and we do metabolomics, and then we compare again. We put into the model, and you can see the red one. So again, using this model, when we do this, this is called test data set. This is called training data sets. Training data sets allow us to give very good one. And when we do the test data set, you can see that again, if you're healthy, we do your metabolomic analysis and we put into this model, again, you're going to be shown to be healthy. But if the patient's one, again, it falls into this particular domain, which show that it is not healthy. Yeah? Of course, there is one dot here that is on the borderline. So yeah, so it's very good actually for us to be able to come up with this model so that we can actually see, to differentiate patient and healthy volunteers. So we wanted to understand why is maltose being a good marker here with a very high sensitivity of 0.1 and specificity of 1 here. It's so sensitive and so specific. So what is maltose? Maltose is basically a simple sugar metabolized by human body. And when we look into publication, we can see that actually people have already published. They said that maltose is usually downregulated in patient samples. That means it should be found to be less in patient sample. Why it is less? Because it is actually being used by the cancer cells to as a food. So when we see, fair enough, it's true in our patients group, our patients breast cancer patient also have been using maltose as a food. That's why when we actually look into the plasma level, so these patients have a low level of maltose here. So again, that's from the earlier slide. So when you do metabolomic analysis, okay, even though you can get very good marker, but make sure you understand why is it up and down, because it's rubbish in and rubbish out, but there must be a science over it. You must see, okay, betul maltose patutnya rendah dalam patient sebab telah digunakan sebagai makanan untuk breast cancer problem. And then same as when we look into hypoxanthine, right? Hypoxanthine is basically a naturally occurring purine derivative. It can be actually salvaged to become nucleotides. So again, we look into some study. Yes, there has been study that shown that there is an upregulation and is related to decrease of this xanthine oxidodidratase in tumor cells. So what we look into is that patients have sort of high hypoxanthine here, because why? This xanthine has been metabolized to become hypoxanthine, right? In this patient, resulting in hypoxanthine to be high here, right? Or the other way that you can look into is actually what happened is xanthine is required to be reduced to hypoxanthine, right? And because there is a decrease of this activity of xanthine oxidodidratase in tumor cells, that's why you can actually see this hypoxanthine to be high in the plasma. So if you actually quantitate hypoxanthine in tumor cells, you probably have higher xanthine but low hypoxanthine, because this xanthine oxidodidratase activity is low in tumor cells. Okay? So again, whether there is high or not or low, you need to know where is it being sampled. Is it being sampled in tissue or plasma, and what are the enzymes that are involved to cause it to be high or low. So there's a lot of metabolism analysis that needs to be understood to make sense of it. So it's never easy in that case. Similarly here again with spinganine. So we need to know how is spinganine being formed, and then where do we actually sample it. But for most of our study, we found that there is a correlation of what we see, right? And same for the rest of the thing. And then again, when we look into breast tissues, as you can see just now for the plasma, we can get very good separation. But with tissue, normal versus this tumor tissue, you can see that there are a lot of metabolites that probably overlaps in the dimension here. What it means, it could be because it's being sampled from the same patient, actually, it's just different side of the tissue that doctor perceive as normal, that it has not been infiltrated with tumor cells as compared to the one that is already have tumor cells. So probably what it means is when there is an overlap like that, what it could mean also is maybe the site where it's normal, they're already being infiltrated with tumor cells. So it's not clean. Is the doctors have not actually cut off the tumor in total, that he has left some cancer cells still in the patient's breast that he considered normal. That's why a lot of times patients after mastectomy, right, after breast removal, their doctors still have to put tamoxifen as a drug to make sure the remaining kind of tumor cells is being cleaned off. So what I want to show here is, yes, metabolomics do not tell lies. It tells you exactly what you did to your patients, is still some metabolites that is being left. Yeah, so very nice kind of thing. But of course, from there you can see what is the differences here, which I think it will take sometimes to explain this, but I will want to skip this, because of time kind of concern. Again, same thing we did for the breast tissue versus normal tissue. We do kind of sensitivity and specificity. As you can see, right, just now for the plasma, you can see very good sensitivity and specificity. But for this tissue-wise, you can see a lot are just 0.7, 0.8, 0.7, 0.8, right? This one is even 0.9. What it could mean also is because, there are contamination of the tissues, yeah, that has not been really clean. And again, when we did a predictive model, again, we actually divide the sample to two sets. One is training, one is test set. And even the training set, we can't actually get very clean kind of separation. Yeah? For this normal tissue, already there are some tumor markers inside here. So what it could mean is actually, yeah, as I said, it's not a clean one. Yeah, it's not that the metabolic model is not good. The metabolic model is good, but somehow the tissue collection is not good. Yeah. Okay, so we also do pathway kind of analysis. And again, majority of the pathway that has been identified in breast cancer is impacted at this arachidonic acid metabolism. Basically, that is an inflammatory pathway, and also vitamin B12 kind of metabolism. Basically vitamin B12 is involved in the production of nucleic acid, right? So when somebody have cancer and is being treated with cancer drug, of course, the synthesis pathway is being perturbated, or being affected. So that's why biotin metabolism is also affected here. Of course, you can see other metabolite pathway. Yeah. So this is the same thing that we did. We can see at the pathway. And then we further divide we further look into, is that a possibility of looking into the metabolism that is different based on the estrogen receptor status of the patients. Yeah? So their patients are being divided into whether it has estrogen receptor negative or positive. And when we divide the patients based on this estrogen receptor positive and negative, you can see that again, there is differences in the metabolites. Yeah, that is found. For example, one example is the spermine that we have been able to find that, this is how we can explain it. Basically, how spermine are involved in the metabolism and is up and down in a different group. Okay, again, we actually look into how we want to explain this. Why is it up and is down? Then we correlate with papers to see that, whether or not people have actually reported similarly with our finding. And we found, yes, there are some supports over what we have found. Yeah. And I have another study on clinical study that is in leukemic patients, children in leukemic. Why I show this study is because in this study, we actually use whole cell. We don't use plasma. So what I want to show here again is that, don't worry, you can use whole cell, you can use plasma, you can use white blood cell also alone, as long as you can actually clean it up properly. Make sure if you want to look into white blood cell, you can really harvest only white blood cell and it's not a mix with a red blood cell. Maybe that will give you a better marker compared to plasma or serum or whatever, right? So, but of course, there are certain way of you treating it. But always remember, for metabolomic, if you're doing global kind of metabolomic, try to do it as simple as possible. Just use chloroform or SCN just to precipitate it, precipitate the proteins. Then after that, you centrifuge it down, and then after that, just straight away inject. Don't have to extract with very complicated kind of approach. Then again, we look into the different metabolites. For example, in this case, for patient was a healthy, well, even though we don't have many subjects, we have only 21 patients and 10 healthy. We can see very good separation of metabolites. Usually, this is the first thing we do. We have to see, is there a difference or not? Then only we look into what are the different metabolites that have been presented. Then you actually can identify what are the metabolites that is different in different group of patients. Then you can actually see, oh, this metabolite is up or low. For example, low in your healthy volunteer but high in patient. Kenapa? So this linoleic acid, right, has been also being found to actually reported similarly by another study. So that is how we make sense of it, yeah? So I'm not going to go through in detail because if there is another group in your group, there is somebody that is interested in ALL, then we can discuss as in how we want to proceed from here. Yeah? So yeah, for other metabolites, again, there's always a reason why. Yeah, it could be, okay, this one is actually high or low in your relapsed patient. Why? Because there is some relationship, whether it comes from the disease itself ataupun it comes from the diet itself. We have to make sense of it. Yeah. And this is another study that we actually did lately in 2020, looking into the methotrexate effect, yeah, the high dose methotrexate in children with leukemia. Again, then we can see how this methotrexate treatment actually affect the metabolism of these patients. Yeah, which pathway is being affected? Again, you will see that methotrexate patients actually, with them, you treat it with methotrexate, this pathway are being affected as well. Yeah? So we find some markers. Next, what we have to do is, again, we have to see whether xanthine is a good marker or not. Because as you can see here, before treatment, xanthine is high. After treatment, xanthine is reduced. And this xanthine reduction has been shown to be a very good marker in our study for patients that do well. So what it could mean is that if patients are treated with methotrexate after this, we want to measure, if you just measure xanthine only, right, you have a VIP score of 2.5, all right, whether or not we can differentiate from patients who will respond versus patients that are not going to respond. And similarly, that is what we did, okay, with a false positive rate, at when it's two positive rate of 0.7, right? It has a false positive rate of 0 point something, yeah. But yeah, the level of cutoff point of this xanthine, we do a relative quantification. The cutoff is 3.75. What it means is at 3.75, all right, we use it as a cutoff to detect if patient have this level, whether it's going to be a relapsed patient or it's not a relapsed patient. Yeah? So this is usually the global metabolomic profile. Then actually we do a MS to detect, to make sure it is truly xanthine that is being there. Yeah. Okay, and then we look into whether or not we will see differences in metabolism if we just look into genotypes. So this is a study that we look into cancer patient, in colorectal cancer patient, and we found that yes, if we actually have enough patients, we can actually see that this different group of patients with regards to UGT1A1 that actually metabolize drugs that is given to this patient, you can actually see metabolism patterns that are different in them actually. Yeah. So, and we can see that, in patients who are having a mutation of this UGT1A1, they have certain markers that are increased. And if they are heterozygote, again, this is another one whereby is moderately upregulated, and this one is actually upregulated in all genotype group, and then this is basically not specific, so you can actually focus in this one. Yeah? If you want to look into the metabolomic effect, that is due to genotypes. And this is some of basically what are the metabolites that has been used, that has been changed up to 16 times different in different patients with different genotype groups. So these are the list of it, and we try to understand each of it by understanding. So I'm not going to go into it. Memang what I'm trying to say is memang there are explanation of what you see if you do stringent kind of statistical analysis. Yeah. And yeah, this is another study that we look into metabolism to characterize diseases, yeah, for to predict myocardial infarction. What we did here is actually we look into orang asli, because orang asli is known to be very healthy. When we collect the subject, they are healthy, but when we look into the metabolomics, we actually found they are not healthy. Yeah? So as I said, when we actually do metabolomic, we actually have to inject the sample 4 times, and when you actually overlap the sample profile, you can see they overlap very well. So basically, this sample means it is very repeatable. Yeah, it is very repeatable. And we also, what we did is that we usually select some of the metabolites, and then we do count the mass differences, yeah, between the different batch of run and also the retention time, and then we find the relative standard deviation. So as a rule of thumb, it should not be more than 20%. Yeah? So then you can say that the metabolites are repeatable. And this is the formula for standard relative standard deviation. Yeah. So yeah, so this is some of the PCA we did, but this is one component. You can see the differences. Yeah. So yeah. Then we follow on with, because actually all these are healthy volunteers actually, yeah. Well, orang asli are healthy, but we look into the compare it with myocardial infarction patients. Then we do again the biomarker kind of analysis and see whether it is high or low. For example, in this case, for this 15S-HETE, the healthy volunteer should have a low kind of level, whereas patient with myocardial infarction should have high. So we are able to actually find markers that denote diseases. Yeah. For again, this one, high in myocardial infarction patients, very high in fact, because that's why it's become red. And then for healthy volunteer, it's low. And then this one, high in healthy and low in patients. So that's why, I mean, recently you find papers that say vitamin D is very important even to protect people from cardiovascular diseases, right? So as you can see from our analysis, you can see for patients with myocardial infarction, you can see their level is low as compared to healthy volunteers. As I said, I don't know whether to believe what we see, but when we compare what we see with what is being published, yes, there is always a correlation. Yeah, so again, we do LCMS, MS just to confirm back whether or not the metabolites that we believe to be a very good metabolite, is it really present or not? Because with the LCMS Q-top, we can do LCMS, MS to confirm that. So, and of course, you can do this pathway analysis, which is very complex. Yeah, then you can denote to see that whether or not these metabolites are being present or not under this metabolism pathway. For example, this metabolite actually belongs to this pathway, and how many metabolite under this pathway is being identified in your samples. Sometimes what happens is that you only manage to identify only one of this marker actually. Then it is not strong. Then your when you do a pathway analysis, it's not impactful enough. That is what it means by impact just now. If, for example, you manage to identify, for example, this glycerol metabolites, metabolism, and these are some of the markers, for example, right? You identify all the markers, then that pathway will be impactful. Okay, for example, this glycerol metabolite is very significant and also is very impactful. That differentiate between myocardial infarction patients as versus healthy volunteers. So maybe we can develop a dipstick or whatever after that. This is our analysis, which well, this is what we get from metabolomics. Okay, I think that's all with metabolomics, but this is just a bit of introduction about I promise, which I would like to introduce before I close my talk. Basically, at I Promise, we do a lot of work based on precision health. What we want to do is we want to empower precision health to build a healthy nation in Malaysia. Similarly, I think the same approach can be adopted by our friends in Indonesia to determine the best care for each individual, to identify disease mutations, to avoid serious side effects from medications, to identify genetic risk, so that we can actually combine pharmacogenomics and metabolomics, right? And at I Promise, we do human genome projects, we do pharmacogenetics project. We also have computational modeling program that do drug discovery, that we are actually in plans to work with Indonesia friends, or metabolomics, protein peptides. We do all this actually. We also one of the center in Malaysia that actually do COVID genome kind of surveillance analysis. And this is what we adopt in our place with regards to our research. As far as possible, we do genome research. We actually do NGS. Then we actually also do metabolomics just to correlate back the relationship between genome and metabolomics, just like what we did with the Orang Asli project. Then we also do drug design, and then because after identifying some targets from here, we actually wanted to see, is there a good drug targets there? Yeah? And we also do mutation analysis. If we identify some new variant, we want to predict first what happened, whether or not the mutation actually caused protein defect or not. Then we actually do functional analysis using zebrafish. We actually can mutate the embryo to see or not whether the protein change actually will cause disease. So this is basically our research platform here at iPromise. And besides doing research, we also serve the industry. Yeah? We actually provide these services. We share with community, science community to share, to actually be able to use what we use here at iPromise. So we do personal genome analysis. We also do bioanalytical analysis. We do metabolomic analysis for our friends, for our customers as well. Then we also do biological, preclinical biological bioefficacy studies. This is what we provide as service. And for personal genome analysis, we actually use NGS as well as array to help our customers to predict their disease risk, to personalize drugs, personalize nutrients, personalize the exercise and things like that. And we also develop some PCR tests in our lab. We actually do pharmacogenetics simple tests using PCR, and also tests to do nutrigenetics analysis. All right, and this is the kind of reports that we actually produce. All right, so with that, I thank you for your kind attention and also for being with me until now. Thank you very much.
Yes, thank you very much indeed, Professor Tef, for your very impressive talks and what amazing works. You did a lot of things, and yeah, I do learn a lot. So I assume that everyone here also learn a lot. Yeah, you say that the metabolomics is not so new, but I think this field is kind of relatively new and we do not have so many experts yet. So I think we're kind of lucky here to can learn from the expert. Okay, so now the floor is open for the discussion. Everyone is allowed to ask questions. For the Zoom participants, please just use the feature raise hands and you can type your question in the chat box. And for the audience in the YouTube, please just write your question in the comment section. I think the committee can help to pick up the question and can help to bring the question here. Okay, so now we are open for discussion. I didn't see anyone raise hands.
I think it's. I think if you don't have a question, it's fine. What we can do is that we can have subsequent session to for example, if you have a study that you want to do, we can actually have subsection to discuss further how we want to do the design of the study. Because I think you have a lot of samples. We want to design the experiment properly from the beginning, right? So if any one of you really wanted to do metabolomic analysis, they can start discussing from the beginning, and then we see how we can collaborate from there. For example, if somebody is working on breast cancer, maybe you don't have to start from scratch. You can pick up from us. We have identified some good markers. So what we can do is that we can actually do quantitative analysis. You can have a small pool to start with, then after that we straight away go to quantitative to quantitate the markers that we already collected. So we don't have to repeat the study. So that is also another way actually.
I see. Okay, thank you very much. But yeah, we have the question in the chat box. So maybe I will just read the question. Hi, Prof. I'm Dr. Daniel here. May I know what's your practice in annotating the metabolites? Do you follow the level of annotation as outlined by Metabolomics Consortium? Okay, thank you, Prof.
Okay, I think the Metabolomic Consortium do have some explanation and definition of what a biomarker has to be, right? And how you want to validate it. That's why for what we have done so far, actually, we have done a preliminary to sort of we have identified biomarkers, but we cannot actually make it into clinical practice until we perform more validation. So I think the collaboration with your counterpart will be good in the sense that if we can actually validate the biomarker using the consortium kind of definition, then it will be good, right? Because at the moment we are using the platform to actually identify biomarkers. using the stringent criteria. For example, if we want to make sort of identify a potential biomarker, it has to be of a sensitivity and specificity of more than 0.7. We have done that. We have done the statistical analysis. We have made sure that it's done. But to allow that to be implemented clinically, we have to do more. We have to do a case control validation study, for example, before it actually can be put up clinically to study. I hope I answer that question. We have to actually follow consortium punya definition in order for us to be able to use that. And later in our own country, yeah, we will also have to follow some medical act also in order to validate the biomarker accordingly, yeah, before we can actually use it in our practice actually.
Okay, thank you so much, Prof. I think, yeah, it answered the question. So is there anyone want to ask? Or maybe not, if not a question like what Professor said, we can discuss some experimental study design. Okay, I'll try to check from the YouTube, and I think we still do not have the questions from the YouTube audience. Okay. Yeah, maybe I can I have a questions?
Sure.
So actually, yeah, my research also about the breast cancer, but I'm usually doing the pathway analysis, usually the genomic information, and I can fully understand that this metabolomics can be very useful for diagnostic tools. We can differentiate like the healthy cells and the cancer cells so that we can know like this from these metabolite profiles that these cells are cancers. But when it comes to therapeutic strategy, I'm still not really can get the full image how this metabolomics information like can manipulate the like a cells fate, cells regulation, and cells pathway in the like a the kind of the genes expressions or something. Yeah.
Okay. Yeah, when you wanted to look into the mechanism of the drugs, it's better you use cell lines because cell lines will be more clean, right? Then you can actually identify the pathways specifically that affected. But when you want to look into, I mean that is for you to understand the mechanism of action. But later you need to, for example, if that thing has been already implemented clinically, patients are basically very complex because they also take herbs, they also take food, they also actually affect how the metabolism actually are different from different patients. So but as we always say, if it's Chinese, they will usually look into holistic. Chinese medicine will be holistic, right? So the metabolomic approach will be very good to actually, if you have two sets of subjects, for example, a subject treated with this medicine and they do well, as versus the other sets of subjects, they take the medicine but they don't do well. If you have good enough of sample size, for example, this group of patients that do very well, but some of them take herbs B, some of them takes herbs C. So if we can actually identify them properly, then we will be able to actually do a very good metabolomic. Then we can isolate out again which particular pathway that are actually solely only affected by the drug of concern. But if we can't actually have a good enough sample size, because we are complex when it comes to patients, the emotion also may actually come. They may have depression, for example, because when it's identified as breast cancer, after going surgery, you can see a lot of this. Sometimes the tyrosine metabolomics also being affected. That's because they are actually having depression, actually. Right. So basically, the metabolomic, I would say, it will actually give you a lot of direction overall about the patient, even the CNS. Also, for example, patients are having depression. You can also differentiate group with depression versus no depression. So what I'm trying to say is, if you want to understand your patient as a whole, all right, you can do metabolomics, because besides knowing the effect of the drugs that you have identified through your cell line, you can also understand exactly what the patient is suffering, because you can see what other pathway are affected. For example, if they are having depression, what actually are happening? I mean, there will be differences that you see in the tyrosine pathway, I believe.
Yeah, I see.
But you must
Thank
get
you.
enough of
Yeah.
because it is now, I think, according to metabolomics publication, each group must be at least minimum 30. So, for example, you have breast cancer patients, right? Inside the same group, you have treatment group and no treatment group, lah, for example. But the treatment group itself is also so heterogeneous. So each of the parameter, if possible, you must have at least 30 subjects.
I see.
So if inside the breast cancer patient, you have 10 different parameters, then it will be 10 times 30, over 300.
I see. Okay. Yeah. Thank you so much for
the
your.
more the better, actually. Yeah.
I see.
Minimum
Okay,
of 30,
thank you.
For minimum of 30, then you can get very good kind of
profile. I see. Okay, thank you, Prof. Actually, we're still waiting the questions or maybe discussion from the audience or participants. Everyone is welcome. Still no.
Dr. Medi, maybe.
Yes. Yes. Dr. Rani, please.
So while waiting for a question from other participants, I'd like to ask two questions. Prof. Teh, thank you first for your very impressive presentations. Impressive presentations. So first question is about the practice. Yeah. Whether you can share us your experience or information about the practice in clinical settings. So I mean that do you have any information regarding the use of the study results from the metabolomic analysis, metabolomic study that then applied in clinical practice settings? And the second question regarding the data collection and data storage that you mentioned before, that one of the important things or crucial things that should be taken care carefully, before conducting the analysis. So maybe you can share us the maybe not the rule of thumb, maybe for data collection and data storage, because maybe depend on the data types, the specimen types, maybe depend on the specimen types, will the treatment for serum, for plasma, for cells, for urine may be different with each other. So maybe in general, how can we treat the samples correctly for collecting and also store the samples? Maybe that's question from me, Prof. Teh. I hope that's clear. Thank you very much.
Yeah, thank you, Dr. Rani. I think the question of whether or not metabolomics has been able to identify a marker that has been translated to clinical use, I say yes. Because of course not from our lab, we didn't manage to get that as yet, because we don't, I think we will have to work harder to be able to get that. As I said, there are actually quite stringent criteria before we put things into clinical practice. So we have to do a lot of validation, and also collaborate very closely with the hospital in order to do that. But yes, like for example, in this prostate cancer, I think there are already biomarkers that has been identified through metabolomics, and now it is being used as a clinical tool to actually help doctors to diagnose how severe is the prostate cancer. Because prostate cancer, I think the problem with prostate cancer is that it's sort of a thin line between having hypertrophy as well as versus cancer of the prostate. So people have worked very hard to try to find, is there a metabolite biomarker that can actually being isolated out to actually represent cancers as versus just hypertrophic inflammation. I think they have found a marker. I can't remember. I think it's sarconsine that actually has been used to actually differentiate between malignant versus cancer, malignant versus benign kind of prostate cancer. Also, yes, there are. Yeah, so I think we already have the know-how. I think if we can work together, I think that is possible actually. Yeah, but for us as after we identify, we have to go through a series of validation as well. We have to actually have the standards, then actually we have to quantitate it. We have to collect more sample to make sure what is a cutoff point of high or low, because I think a lot of these markers are actually endogenous biomarker. It is actually inside our serum or plasma, but it's just the level is high. When it's high, then it denotes cancers, things like that. So we need to have a lot of samples to be able to do that validation, which I believe collaboration with your counterpart, definitely we can get something out. Yeah. And with regards to your storage, that storage of samples, I think that again very critical and very sort of very good question, because if you want to collaborate, for example, between I Promise with Indonesia, how we want to make sure the sample that is stored by you is still okay, right? So there's two ways to look into it. I would say the best approach is actually once you collect the sample, once you actually have sort of, if you want plasma, you have already spin it down to take the plasma, then you store it straight away, you freeze it with liquid nitrogen, freeze it so there is no more chemical reactions that happens. And on transportation, yeah, you still on liquid nitrogen, then you put it into your freezer -80 if possible. Right, that one would be okay. Same as for all kind of cells, tissue sama juga. If you can actually collect it and straight away freeze in liquid nitrogen, you already freeze the reaction, so there is no going to be any more reactions, right? So if for example, if you want to collaborate with us, maybe you want to send sample here, I will think it's better you extract out the metabolite, freeze the metabolites, and you post the metabolites rather than putting the sample still in the plasma or serum, because inside the plasma or serum there are still some enzymes. So when you freeze it, they may actually slow down activity, so metabolism still happening. But if you actually have purified the sample, remove all the enzyme, no more proteins, enzyme inside there, it's only merely the metabolite. You freeze it, probably that time when you post it on ice, or when you put it on dry ice, that should be okay. Yeah, that is one. The other thing is, if that is going to happen, what we can claim is that we are looking into stable metabolites. If the sample is, if the metabolite we identify is so easily being metabolized, it is not stable. No point we actually get an unstable marker to be a clinical marker, because being in clinical later, a nurse take a sample, they forget and they put it somewhere. Okay, they put it somewhere before they send it to your lab. By the time they send it to the lab, if the marker is not stable, it's already degraded. So we treat that as a marker. Then when our diagnosis come out, we say, oh, patient is healthy, they don't have the marker. Padahal it's because the nurse has actually left the sample outside, it's unstable and then it's gone. So for me, for clinical diagnosis, it's better we look into stable metabolites, right? It's best if we leave it there for 2-3 days, it's still there, then it's okay, fine, it will be a very good clinical sample.
Yeah.
It's just, yeah. So for clinical, it's like that. But for you to do experiment, like for example, you want to understand the mechanism, probably you want to know every single metabolite. That one you can do it in the lab. That one is in your control. The one you can identify all kind of metabolites, stable, unstable, put okay, because you even want to look into the unstable one so that you can actually detect, actually it's through this unstable pathway that it actually take this pathway for the mechanism of action. So it depends on our purposes lah. Yeah. I hope that answer your question.
Oh, very clear, thank you. So it depends on our purpose. So first, for knowing the mechanism, we have to treat the samples very careful. But in other case, if you want to use that as a clinical biomarker, clinical marker, the new clinical marker, so we are looking for a stable marker. It means that we can treat the samples as usual. At least we have same treatment for all the samples, maybe, Prof. Teh, yeah?
Yes.
Maybe we can, and also, we give information about how we collect and store the sample, and we are looking the one that stable and has significant results compared to the others. Thank you, Prof. Thank you very much for your explanation. Thank you.
Okay, thank you for the previous discussion, Dr. Rani and Prof. Teh. Maybe we're still waiting if from the audience or participants, or maybe I could, actually there's some people try to reach me to give their question, and maybe it's very too technical. She said that how much the weight we need, like we have if for example we have the breast cancer cell lines, it's like there is some standard, like how much the cells we need, like to extract the metabolites, and actually now we have it on the paraffin. So is it still like good enough, like if we want to extract the metabolites from the cells?
Okay, I think when the machine become more sensitive, I mean, maybe when we do it like 10 years ago, the machine has not been very sensitive, so you need a larger amount of samples. But when the machine has been adjusted to be more sensitive, for example, then we can use less amount of samples, right? For example, in terms of cell culture, before this we work on 10 power of 6, at least 10 power of 6, because when we have cell lines, you actually we usually work on 10 power of 6 to get enough kind of markers. But cell culture is okay because you can actually just grow them, right? What is critical is the clinical sample. For example, those sample in paraffin. Again, as I said, for clinical sample, we do with what we have. For example, if we can harvest from paraffin, but make sure we still need to know the date, as in, when is it actually being fixed on the paraffin. It probably will be different compared to after 1 year paraffin as versus 10 years after paraffin. So that kind of metabolites way, I mean, it still may be different. Yeah, so we can still try to harvest some of it. I think you can still see some pictures. As I said, we look for, markers that is sort of, if it's a biomarker that to denote certain diseases, it's fine. We don't have to understand everything about the disease. We are not looking into the mechanism of action. So it's fine for as long as we can find a very stable marker that actually differentiate disease A versus no disease. That is good enough, right? Yeah. So we can still try paraffin.
We still can do even very with a small amount, and even we already storage it in the paraffin. Okay.
Actually, last time our there's one Professor Rudolf Grimm. They actually work on glycans from mummies. Mummies that have thousands of years, right? They actually go and get the sample and look into the glyconomics. They can find differences.
I see.
Yeah. So
very interesting.
So there may be some glycans that are very stable in terms of its bonding, the formation. It will not degrade. Then that is also interesting actually.
Okay, thank you so much for it's very clear answer. I hope the audience also can feel the answer is very clear. Okay, so I also get one question from the audience, but they reach me through the personal message. So, Professor, do you have the challenge to study metabolomics in clinical setting using pediatric respondents?
Do I have?
Using pediatric respondents, not sure. Maybe when you collect the samples from the pediatrics, do you have you ever experienced?
Our leukemia subject basically about not really kids per se, but it's children, not neonates. Neonates we have not done, but I think it should not be a problem because we can still do, because like those children or neonates with inborn error, they are actually done with this metabolomics as well. But I personally have not done on inborn error metabolism, because if you want to do that as a diagnosis, then you need to have a machine that only do that, because I think we would actually caution on basically we cannot mix many studies using the same machine if we want to do a diagnosis for inborn error metabolism, because you may miss some of the metabolites or something.
I see. Okay, thank you very much once again. For your explanation. Hope it answers. Okay, yeah, give the insightful discussion. So now we come to the almost the end of our webinar. But before that, our committee would like to give the appreciation for Professor Teh. So the committee will give the certificate, which will be given by Dr. Rani virtually. So I will welcome Dr. Rani to give the e-certificate to Professor Teh. Please.
Thank you so much. Thank you.
Yeah, Prof. Teh. Thank you very much.
Thank you very much.
Okay, maybe we like want to take a picture. The committee can help to guide for the picture for a photo.
Yes, we want to take a picture, Dr. Rani and Prof. Teh. And count 3, 1, 2, 3. Thank you.
Bu Citra, we have Prof. Ari here.
Prof. Ari, Prof. Ari still have guests, Bu Rani. So as you.
Sorry. Okay.
Okay, thank you, Professor Teleke, for this opportunity to share your experience to us. Yeah.
Thank you, Prof. Yes, thank you for inviting me and give me.
I hope we can collaborate with this type of research in future. Yeah.
Definitely.
Yeah.
Yes. Thank you.
Okay, thank you once again. Yeah, for Prof. Ari, Dr. Rani, and Professor Teh. Okay, so many thanks to Dr. Teh, Professor Teh, Dr. Rani, and Prof. Ari. So, okay, we come to the end of our event. Once again, I would remind the participants and audience to fill out the attendance form and also the satisfactory survey form, and also the certificate will be given to the participant who did a post test with the minimum score of 80. So, okay, please later check in the chat box to about fill out the attendance, the survey, and also the post test. Okay, finally we come to the end of this event. May all of us get the value and the benefits from this today's webinar. Thank you very much for your kind attention and participation in this webinar. Yeah, I'm Medu Putri signing off and see you in the next event. Assalamualaikum warahmatullahi wabarakatuh. Yeah, thank you.
Thank you, Prof. Teh. Bye.
Thank you.
Prof. Teh.